Welcome back. We were off for about a week, right? With Easter Monday, we didn't have any class. So hopefully you had an opportunity to chat with the family, even if you didn't get a chance to spend some time with them physically, play FaceTime or something of that nature. We were covering Chapter 8. So to remind you about where we were, right, we were covering synaptic plasticity, the idea that changes in the activity at a synapse can lead to changes in the strength of that synapse, both short and long term. And so we had covered this organism called the Aplysia. It's a sea slug, and it was a great early model system for those who were studying synaptic plasticity. And we had talked about the habituation response, right? That over time, the response to a stimuli decreases. We talked about the methods or mechanisms that underlie that. More recently, we were talking about sensitization, okay, this effect of strengthening one synapse by activating another synapse, okay? So I'm going to bounce back to this picture quickly. Right, and so the idea behind sensitization is that we were going to trigger this sensory neuron here, right, to generate an action potential by giving a tail shock here, okay? And we were paying attention to what was happening at this particular synapse, right here, right, between the modulatory interneuron, right, which is activated by the sensory neuron. So this modulatory interneuron is carrying information because it's been stimulated, and then there's going to be a synaptic event here, right? That's going to involve the modulatory interneuron onto the presynaptic terminals of this sensory neuron that's involved in the siphon touch response, okay? So note that this modulatory interneuron does not directly innervate the motor neuron. Okay, this does not occur. Instead, the modulatory interneuron innervates the presynaptic terminal of this second sensory neuron here, the one that's going to be activated when the siphon is touched. Okay, so we went through the mechanisms of that, and just to give you a refresher on that, right, we talked about how it's protein kinase A dependent, that here's the modulatory interneuron, right? They call it a facilitatory interneuron here. And you can see that this synapse is not between the facilitatory or modulatory interneuron and the motor neuron, right? Instead, it's at the presynaptic terminal. Like we said, it's at the presynaptic terminal. I'm going to jump back here, of this sensory neuron. Okay, so this, I'm going to circle where we're talking about, right? We're talking about this synapse right here, initially. And so this modulatory interneuron is releasing neurotransmitter, which is activating a G protein, which leads to the eventual release of PKA, or the activation of PKA, I should say. And the end result of PKA activation is essentially more neurotransmitter release. Okay, so we can talk about that, or I talked about that in the last video, but the end result of this, how this synapse gets sensitized, is that, right, this is the sensory neuron that responds to siphon touch. And the idea being more neurotransmitter will be released due to the actions of this facilitatory interneuron here. Okay, so if you have questions on this part, or you want to talk about any of the mechanisms, the last video covered it. Otherwise, let me know and we can have a conversation about that. Okay. However, this effect that we see, right, this sensitization effect, really lasts for only about an hour. But there's another mechanism that can prolong sensitization, and that's what we're going to look at next. So let's discuss long-term sensitization. And we're going to look at this in the same synapse as we were just looking at, right? So here's the modulatory / facilitatory interneuron. Again, the synapse is not directly on the motor neuron. The synapse is on the sensory neuron, right? That which is responding to siphon touch. And this motor neuron here, right, the eventual result is if this motor neuron generates action potentials, it leads to a gill contraction. Right, because it's going to innervate that particular muscle of the gill. And so again, what we're going to see are synapses that occur on the presynaptic terminals of this sensory neuron here, okay? So where do we find the receptors for whatever the facilitatory interneuron is releasing? Not on the motor neuron, not on the dendrites, but there are actual receptors that we'll talk about that are on the presynaptic terminals. So typically when we refer to anything that's long-term with respect to plasticity, we're going to see changes in either gene changes in gene transcription or typically protein production, right? That's the translation portion. And so this long-term sensitization will be no different, okay? We're going to start by activating the same system that we activated before in the shorter-term sensitization, where the facilitatory interneuron releases serotonin. So there's a similar release of serotonin. Right? And this serotonin released by the facilitatory interneuron has serotonin receptors, G-protein coupled receptors, on the presynaptic terminal. So maybe we'll say that the GPCRs on the presynaptic terminal got away from me there, but that's presynaptic terminal of this siphon touch sensory neuron, okay? Now, once those G-protein coupled receptors are activated, the end result is protein kinase A activity. So, protein kinase A is now active. And again, I think it's important to go through the process, right, that you've got an enzyme here, adenylyl cyclase. Adenylyl cyclase converts ATP to cyclic AMP. Cyclic AMP binds to the regulatory subunits and allows the catalytic subunits to be active. That's all stuff that we talked about in chapter 6, 7. In any case, right, one of the targets of PKA we talked about last time, potassium channels, right, with the short-term sensitization. With this case, one of the targets of PKA is CREB. The transcription factor CREB, and CREB becomes phosphorylated. And now can work as a transcription factor, so they show that the phosphorylated CREB is here. And what we see are changes in transcription levels, okay? Followed by changes in translation, so we get increased protein production. The protein that gets produced is one called ubiquitin hydrolase. So ubiquitin hydrolase, this enzyme is made. So its concentration goes way up in the cell, right? Without PKA, there's no phosphorylated CREB. Without any phosphorylated CREB, the gene for ubiquitin hydrolase is inactive. But now this gene can be used to produce mRNA and produce a protein, right? The ubiquitin hydrolase protein. What the UH does is it degrades the regulatory subunit of PKA. So this is different than binding the regulatory subunit and releasing it, right? So cyclic AMP, we talked about how cyclic AMP binds to the regulatory subunit and that releases the catalytic subunits. Well, in this case, the regulatory portion is just degraded. It's completely degraded. Okay, well, without any regulatory subunit, we use this term constitutively active, meaning that pKa is going to be active all the time. So now pKa is constitutive. This is a good biology word. You'll see this show up a lot. Constitutively active. Which means that there's no regulation on the pKa anymore. Okay? In this case, pKa will continue to do what we've known it to do, which is change the function of potassium channels. So we can write that pKa will now target, like we've seen before, right, is constantly targeting and inhibiting potassium channels. Right, and like we saw previously, inhibition of potassium channels prolongs the action potential, allows more calcium to come in, and we get increased neurotransmitter release. The other thing that happens here, so once Krebs is phosphorylated, we said that ubiquitin hydrolase is produced. The other response that's Krebs dependent, another target of Krebs, right, so I'm running out of room, but I'll draw Krebs phosphorylation here, leads to activation of another transcription factor called CEBP. So CEBP is also a transcription factor, as is Krebs, and it's going to induce the production of mRNA from different genes than Krebs does. So they have slightly different targets, but they're both going to induce transcription. And while we don't have a great idea of the specific genes that CEBP regulates, what we do know is that what we see after it's activated is a change in the structural architecture of the synapse. So actually we see additional synaptic terminals added. So increased synaptic terminals. All right, so you could imagine you've got this one synaptic terminal here, but potentially with the induction of CEBP, and now it's going to work to activate genes, we might see more synaptic terminals coming off of this branch. Okay, and so you could imagine that this would also, if this synaptic terminal is going to synapse on that motor neuron, also going to strengthen the synapse. These are long-term structural changes, right? This isn't like regulation of an ion channel, which can be flipped on or flipped off. We see changes, like I said, in structure. We see changes in the presence of protein, or at least the form protein takes, like with pKa not having a regulatory subunit anymore. And so either of these, right, are thought of as long-term mechanisms which will persist for many, many days.